So now we're going to see how algae can work on the balance of our body. He got his doctorate in biochemistry from the University of Clermont-Ferrand. He then went to the United States for 3 years to do his postdoctorate in Madison, in Wisconsin. Then he came back to France to work at INRA as an engineer and researcher. His work looks at the bioactivity of polysaccharides, marine polysaccharides in animal health. Ladies and gentlemen, please welcome and applaud Professor Mustapha Berri. Professor, you have the floor.
For inviting me and giving me this opportunity to give this talk and to present a summary of an update of the biological activities of polysaccharides that were extracted from marine algae. And the second part of my talk will be dedicated to the work or the preliminary data that we obtained during a collaboration work that we performed with the All-Mix group. But before starting the lecture, I would just to introduce myself as engineer and not professor. I would like to be, but maybe it's too late. Anyway, so I think during this lecture we will catch up the time that we lost this morning, because as the former speakers talk about the intestine and how this organ, which can be considered as tertiary lymphoid organ, and how Bernard explained also how the polysaccharides are formed and their biological activities on human. So maybe we will catch up the time and we will go for lunch very quickly. Please let me introduce our institution. Like Hervé, I am a part of the National Institute of Agronomic Research, which is over 8,000 people working on different areas such as agronomic research, agriculture, nutrition, and environment. And this institute is presented by over 17 centers all over the country, that, as you see in this, oh, I'm sorry. How can I go back? It's like this. And the pointer is on the green, with the green, right? Oh, no, my God. OK, so this one. No. Ah, voilà. Bon, on va aller, ça c'est devant, et c'est ça. So our center is located in Tours. It's 250 kilometers south of Paris. And you can see, and Hervé is coming from Jouy-en-Josas, so we are neighbor. And there is other centers in Clermont-Ferrand where I had my PhD, like it was introduced. And there is also a center in French Guiana and in Antilles. And this region is also, it's also known by all these beautiful castles that you can see, like Azel Rideau, Chenonceau, Ovilandry, and we have also good food. So may I suggest to Hervé Balusson and All-Mix organizer to go just 150 kilometers further north, just to and organize this meeting for the next year. Bon. So, as I said, I belong to this research unit, which is dedicated to study the host pathogen interaction, such as bacteria, viruses, and parasites. And the main research programs are dedicated to characterize pathogens, their diversity, their virulence factors, and how they resist to drugs like antibiotics. And we also study trying to understand the molecular and cellular mechanisms of host pathogen interactions. We also trying to analyze the host immune response toward microbes. Why are we doing all this? The main goal is to evaluate new prophylactic strategies and to find some other molecule and test other molecule as to replace antibiotics. You know that the main issue in the next century is the resistance of bacteria to antibiotics. So I choose this picture just to show you how, if you overfeed your animal with antibiotics as growth promoter, your animal will be protected against known strain of bacterial infection, and this bacteria may be mutated and be resistant to the antibiotics, and this antibiotic resistance could be transferred to a human, and a human will harbor some bacteria that will be resistant to antibiotics. So that's why the European Union voted a regulation to ban the use of antibiotics as a growth promoter, and it entered into effect on January 2006. So what should we do for the next time? So the question is, are there really any alternative to antibiotic use? And the answer, I would say, yes. So the first alternative is vaccination. And the vaccination is very important, because if you vaccinated the mother will be protected against infectious disease. And this mother is able to transmit this protection to their neonate by maternal immunity through milk suckling. The other alternative that can be considered is by using probiotics, which is it's a harmless lactic bacteria that we can use as a feed additive. And the other alternative that we can consider also is prebiotics. This prebiotic is mainly oligosaccharides or polysaccharides. So if we focus our attention on using polysaccharides, so we need a plant that will grow very fast, and a plant that not need rainwater, and a plant that are able to produce a high amount of biomass and biological components. So this plant could be considered as algae. So I mean, since we have working with Olmos Group, I knew a lot of things about algae, and maybe you know better than me about this plant. So the algae is the fastest growing plant organism in nature, and it has the ability to convert a large amount of carbon dioxide into oxygen. And today, I know that algae still produce over 70% of the Earth's oxygen. It has the ability to triple or quadruple their biomass every day, more than corn and beans and soybean. And as Bernard said, this plant also produce active components like lipid, protein, vitamins, minerals, fibers, and polysaccharides. And these polysaccharides are considered the bioactive component in algae. I am not going into the details about the structure of sulfated polysaccharide, because Bernard has said everything about that. But just keep in mind that the ulvan is a sulfated polysaccharide that was extracted from the green algae, and carrageenan it's from red algae, and fucoidan it's from a brown algae. And we can go further, because Bernard said a lot of things regarding the structure of this sulfated polysaccharide. And he said also, he said also that this sulfated polysaccharide has a lot of health benefits. It has the power, it has the ability to prevent the blood coagulation. And it has also the ability as antioxidant. And in vitro study on tumor cells, like you will see further in other slide, mention that this sulfated polysaccharide has anti-cancer activity. It stimulated the dermal and fibroblast proliferation, and it has also the ability to inhibit the growth of bacteria, viruses, as well as fungus. And also, as Bernard said, it has also the capacity to modulate the human response toward human stimulation or anti-inflammatory action. So if this sulfated polysaccharide has all this health benefit, so I mean they will have a potential industrial application in functional foods, in nutrition, in drug medication industry, as well as in cosmetic. So if I put the antimicrobial activity and the modulation of the immune response in different color than this one, is because the coming slides will be dedicated to those two activities. So let's go further. I just summarize in this table the ability of sulfated polysaccharide here from red algae, brown algae, or green algae to inhibit the proliferation of viruses. And the viruses that were tested in these studies are a human herpes virus, as well as human papillomavirus and HIV and HIV viruses as well. So, and how things work, so how the sulfated polysaccharide act on viruses. Probably there is different ways how this polysaccharide act. The marine sulfated polysaccharide may act directly as antiviral action on viruses. It has also the ability to inhibit the transcription and the virus replication as well. The other studies show that the viral absorption and internalization of the virus into the cell was inhibited by marine sulfated polysaccharides. The marine sulfated polysaccharide has also the ability to improve the host antiviral immune response and reduce the mortality rate of animal. And there is another study that showed that the inhibition that the marine sulfate polysaccharide has inhibit the fusion the viruses with this lymphocyte, which are CD4 lymphocytes. So, do marine polysaccharides have any antibacterial activity? The answer is yes. So, when I summarize in these slides, couple studies pardon. Pardon. In this slide, I summarized the couple studies that have been done regarding the antibacterial activity of marine polysaccharides, and these activities so you can see that the activity of marine polysaccharide was performed on gram-positive and gram-negative bacteria, and these studies came from all over the world, and so the marine polysaccharide sulfated were tested against bacteria. This bacteria has has a high impact on animal health, and they can also be transmitted from animal to human. This bacteria included Staphylococcus aureus and Salmonella typhimurium, E. coli, qu'at that we can find in pig, Pseudomonas, as well as Klebsiella pneumoniae. So I am not going into the details into the human system, but you have just to know that human system can be divided into two parts, and all these things were explained in the previous lectures. There is an innate natural response and adaptive specific response also. The innate natural response, I mean, the different actors are neutrophils, monocytes, macrophages, and natural killer cells, as well as dendritic cells. The process that these actors will do is phagocytosis, antigen presentation, and cytokine production. The adaptive specific response is divided in two parts, which are cell-mediated immunity characterized by Th1 cells and Th2 cells. And these Th1, Th2 cells are able to produce cytokines. Th1 activated macrophages, and T cells can lyse the infected cells. And Th2 B cell activations will lead the B cells are involved in B cell activation and by inducing antibody response. So for the next slides that you will see, the studies that I will present to you this morning is regarding how the marine sulfated polysaccharide can stimulate the dendritic cells. And the second part is how this marine sulfated polysaccharide can stimulate the macrophages. And if this happens, if it is true, it means that the marine sulfated polysaccharide can be used as adjuvant in vaccine stimulation in order to enhance the immune response of animal or of a human being as well. So the first study regarding the immunomodulatory effect of marine sulfated polysaccharides, the bone marrow cells were activated with GM-CSF. This is how we do in the laboratory, and the dendritic cells, they were stimulated with 50 microgram per mil with fucoidan, and the positive control is anti-CD40 antibody. So, and the analysis were performed by FACS, and this is dendritic cell, as you can see in this slide, and this is the P6 well where these cells are cultured, and the fucoidan at this concentration is mixed with the cultural media. And the first result that the authors were trying to see if the fucoidan has any cytotoxic effect on dendritic cell proliferation. And you may see here that the percent of dendritic cell viability does not change when the cells are treated with the marine sulfated polysaccharide. And the dendritic cells, in order that dendritic cells should be very active, normally these cells must express which we call maturation markers, which are MHC1 and MHC2, CD45, and CD86. They are just protein on the surface of dendritic cells. And if you can see this FACS analysis and this figure that show the same thing than this, you can see that with the black bar, the cells when they are treated with the fucoidan, and you can see that the high expression of all these maturations factors when the cells are treated, comparing to negative control, which is in white here. So the message here is that the fucoidan enhances the expression of the maturation markers on the surface of dendritic cells. So, but it's not enough, because dendritic cell must express the maturation marker on their surface, but also they have also to express cytokine, and this is the second thing that the authors trying to see. They took the bone marrow cells that were activated with GEM-CSF as for the first experiment, and the focoidan at the same concentration, and the positive control was the same, and they did an ELISA just to check if there is a protein of NO, TNF alpha, and EL12. As you see, when the dendritic cells are treated with focoidan, they express a high level of all these three proteins, comparing to the control positive, and unlike the negative control of the cells that were not treated at all. Let's move further. So, as I say before, this is the experiment, another experiment which has been done on mice macrophages. They are very known by the name of Row 264.7. They are most of the studies regarding the immunostimulation of sulfated polysaccharide has been done on macrophages. So the experiment here is, this is macrophages, and this is P24, well, where the macrophages were cultured and treated with sulfated polysaccharide. And the analysis that has been done is they choose some immunoresponse markers, and they did RT-qPCR analysis just to show if there is any expression at messenger RNA level. So the answer is yes. So you can see on these figures that, let's go back. Ça va vite. Voilà. And you can, and they check TNF alpha, EL6, EL10, and COX-2. And before to do the experiment, what they did is they took the crude extract and separated into different fractions. They were named here. This is the crude extract, and the fractions were named F1, F2, and F3. And you can see that the fractions that were the most active was F1 and F2, and it corresponded to the low molecular weight sulfated polysaccharides. So the conclusion is that the low molecular weight polysaccharide with molecular weight under 15 kD of ulvan stimulate the expression of various cytokines and might be beneficial for immunostimulation. So, as we have seen that the marine sulfate polysaccharide can stimulate the dendritic cells and can stimulate also macrophages. So the question, could we use this marine sulfated polysaccharide as an adjuvant in a vaccine? And the answer is yes. This is a study that has been done on mice. So please take a look over here. This is mice that were vaccinated with 10 microgram with a human papillomavirus E7 peptide vaccine, which I called E7, in combination with 10 microgram of carrageenan with a boost 7 days later. This is the facts. This is the facts analysis that has been done, and you can see that the combination of the vaccine and the marine sulfate polysaccharide has the population of lymphocytes that express CD8 that are CD8 positive and produce interferon gamma. These cells are able to destroy the tumored cells. This is what we will see in the next slide. Is this? Here we go. So here, the mice were vaccinated and injected with tumor murine cells TC1. These cells are long epithelial cells that were transformed with E7 and E6 antigen of a human papillomavirus and race oncogen. And this is the experiment. So they try to see the number of the mice that were free of the tumor. And you can see when the mice are treated with a combination of this E7 and the carrageenan, this rate of free mice, of tumor-free mice, is very high, comparing to the mice that were treated only with E7 or the carrageenan alone. What they try to show also in this experiment is they monitor the mouse survival, the mice survival also, and you can see in this figure that the survival rate of the mice increased when the mice were treated with the antigen and the carrageenan. So the conclusion of this study is that the co-administration of carrageenan enhances the specific CD8 positive T cells response generated by E7 peptide vaccination, and the co-administration of the peptide and carrageenan enhances the protective effect generated by E7 peptide vaccination. So this experiment is the author, they wanted to know what is the mechanism which is involved in this protection. And the first thing is, what they did is they use mice which are knocked out on TLR4 receptors. This TLR4 receptors is expressed, it's talc-like receptance 4, which is expressed on the surface of the cells. And you can see that when the mice are vaccinated and treated with carrageenan, the mice that not express the TLR4, the rate of CD8 positive interferon gamma positive cells decrease, as you can see, comparing to the wild type. So the conclusion is the carrageenan leads to the enhancement of immune response generated by E7 peptide vaccination through TLR4 activation pathway. So does the marine cephalopolysaccharide have any effect on animal in vivo on animals? So this is two experiments that have been two experiments that have been done. This one was they try to investigate if the seaweeds sulfate polysaccharide has an effect on the performance of the pig selected bacteria population and cytokine expression. So there is a low effect on animal performance, and the treatment of the animals with the seaweeds reduce the enterobacteria population and low effect on lactobacillus and bifidobacteria. But it has an immunomodulatory effect with downregulation of EL alpha, TNF alpha, and EL17 in the colon. And this study was done to investigate if there is any effect when you give the marine sulfate polysaccharides to the mother, and if it has any effect on piglet health. And you can see this is the result that were obtained. This is the summary of 3 or 4 papers done by the Irish laboratory. There is a high IgA and IgG in sow colostrum. They saw also that there is no effect on IgA, IgG, and IgM in sow milk as well, but it has a high IgG in serum of piglet that suckle seaweeds supplemented sow, and no apparent effect on intestinal morphology of the piglet suckling marine sulfated polysaccharides. So in few slides now, I would like to summarize the data that we obtained recently during this collaboration with the Olmix group. So the algae were obtained in Plessis-les-Grèves, close to Saint-Brieuc. This Olmix green algae, this is how this sulfated polysaccharide looks like when we receive it in laboratory, and ça marche plus. So this the marine sulfate polysaccharides that we obtained from Olmix group was dissolved in water to do the antimicrobial activity, and it was also dissolved in culture media to do the stimulation of the human response mediator. It was sterilized, and we prepare different concentration, and we check if this polysaccharide has any effect to inhibit the growth of the bacteria or to stimulate the human response. And so we tested 5 bacteria that you can see here, and this is bacteria that we can find in livestock production, and this is the bioscreen that showed that sulfate polysaccharide, it inhibits the growth of Listeria. If this is in red spot, it's the control of bacteria that was not treated. And if you treat the bacteria with the Olmix sulfated polysaccharides, the exponential phase of the bacteria growth decrease. And it's more, we can see it more here with the Staphylococcus aureus, where the bacteria growth is completely inhibited when we use the concentration of MSP at 2 or between 2 or 8 milligram per mil. So this is what we found. We determine the minimum concentration that inhibit the growth of bacteria, and it's for Staphylococcus aureus it's between 1.4 and 3.9, and for Listeria is between 31.3 and 62.6, and it's this MIC, it's similar for the 3 others bacteria. So the second part of the experiment that we have done with the Olmix group is trying to know if this polysaccharide has any effect to stimulate the intestinal epithelial cells and stimulate the cytokine production. So we use IPEC cells, which are pig epithelial cells. They were differentiated and treated with sulfated polysaccharides. We purified the RNA, and we did cytokine expression analysis. And why we use the epithelial cells, intestinal epithelial cells, is because all you hear from the former speakers that the intestinal epithelial cells is very important for the host protection, and these epithelial cells express these receptors, talk-like receptors, TLR4, TLR2, or TLR5 and 10, and the stimulation of this TLR will lead to what we call the signaling pathways and the expression of cytokines and chemokines through NF-kappa-B, for example. So let's go to the result. So what we have seen during this work is that the marine cephalopolysaccharide that we obtained from oil mix has an effect on the expression of TNF-alpha, EL1-alpha, and CCL20 chemokine, which is very important for dendritic cells recruitment and EL8. This is a table summarizing the expression fold change and all the cytokines that have been tested during our study. And as for the previous study that has been done on mice, we also wanted to know if the marine polysaccharide can stimulate the immune response through TLR4. So we did the experiment with the human embryonic kidney cells that stably express TLR4, and as you can see here, that this polysulfated polysaccharide at 0.1% can stimulate TLR4. And we did not obtain any stimulation with TLR5, node 1 or node 2 as well. These are two slides of summary. And the conclusion is that the sulfated polysaccharides that were extracted from the oil mixed green algae is able to recognize TLR4 on the epithelial cells. This is what we have done. And let's go. And when the polysaccharide recognizes or bind to TLR4, it induces the expression of all these cytokines. These cytokines are very important for the host protection and for the anti-inflammatory response of the intestine as well. So, and just to finish this lecture is that the algae and microbial polysaccharides, both they bind to the common surface receptors and induce similar immunomodulatory response. So here, the polysaccharides, this is a summary of most of the in vitro data that was performed on macrophages and dendritic cells. And just to give you a message how things will work if the polysaccharide has any stimulation effect of the immune response of the host. So you can see here that the polysaccharides will recognize a receptor which is TLR. It could be dectin or SER3 or mannose receptors, and these macrophages will be activated and split in two macrophages, M1 and M2. This one will induce cytotoxicity and destroy the pathogens and infected cells. And this other side, and it's a matter of balance here, and this other side will, the macrophages here, the M2 cells, they will express some cytokines that are involved in the human response and tissue repair. And the next thing is, to summarize the immunostimulation effect, is the polysaccharide can also recognize the same receptors on dendritic cells, which are the antigen-presenting cell, and this dendritic cell will give the information to lymphocyte and Th cells to produce cytokines. And these all cytokines, they will do what? They will improve the mucosal immune response at homeostasis, but also they will protect the organism against pathogen infection. And as I say also, this polysaccharide may have an action to boost the immune response as an adjuvant or or vaccine. And the last thing that I will mention before to finish this lecture is that the human cell's response to algae sulfated polysaccharide mimics the human response to microbes. So this is the how to say, a notion of mimotap that we can maybe talk about, and if you have any question like on this. So just before to finish this lecture, I would like to thank all the Olmix group, Hervé Demey, Mathieu, Pi, and Marion, and Olivier. And I would like also to thank my colleagues at the INRA, at Nouzy, Michel Olivier, Cindy, Emmanuel, Isabelle, and Sébastien. And I don't want not to forget Henri Salmon, because Henri Salmon, who is in this audience, who was our previous lab director, and he was the guy who initiated this collaboration with the Olmix group. So, and thank you for BPI France and Easy Ulvan projects, and thank you for your attention.
Merci. Merci.